So we'll do a review of the autorhythmic and contractile cells, take a break, come back and do cardiac cycle, take a break, take the lab quiz, give you some study time, take the lab quiz, and then I'll pass back the exams, and if you leave early, that's fine because you've got that Physio X to do at home. You can use some of the lab time for that. Okay? Now, what locations do we find autorhythmic cells? Because we're going to be talking about ECG today as well before we get to the cardiac cycle. So what locations do we find autorhythmic cells?
SA node.
Okay, SA node, the sinoatrial node, AV node, bundle of His or AV bundle. Either one is fine. Bundle branches and conduction myofibers. Without neural intervention, without the binding of neurotransmitters, acetylcholine or norepinephrine, the SA node is going to depolarize approximately 100 times a minute. So it's usually the fastest one, and it overrides, causes the AV nodes to depolarize at the same rate. The AV node, if the SA node isn't working and we're dependent on the AV node, it's going to depolarize approximately 40 to 60 times a minute. And the AV bundle, about 40. These lower individual contractile cells, what we're going to talk about next. So individual contractile cells in the ventricles or in the atrium are approximately 15 times a minute. And they're, of course, not coordinated. All right? So when we look at our autorhythmic, I asked you to be able to compare it to the action potentials of skeletal and heart contraction cells. So that's the autorhythmic cells. And what we're going to look at again today will be the cardiac contractile cells. And then we compared it to skeletal muscle. And remember, for each of these, I want you to be able to identify the potentials. So we have resting membrane potentials, graded potentials, action potentials. Okay? This is also a graded potential, because it can change the rate at which it changes, goes up or down. And then be able to identify what channels are contributing to that component. So that's what I want to go back over with you today for the cardiac cells, starting with the autorhythmic ones. So when we talked about the autorhythmic, let me draw this a little bit larger here. Do the autorhythmic cells have any resting membrane potential?
No.
No. All right, so they have a graded potential. Is it a graded potential because we can change it by the amount of ligand chemicals that bind to channels or not? Yeah, we can make it steeper or we can make it more shallow. All right, so if we're using the sympathetic nervous system, then we have a steeper. Osmotic ions flow into the cell faster, takes less time. Or if we're using parasympathetic, we can have it take longer. Okay? So PS for parasympathetic, S for sympathetic effects. So that's the graded potential. Now, what types of channels? Can I erase this list over here on the board? You guys have that? And may I erase these 3 types here? You have them from Friday, I know. Okay, leaving this up here, I want to redraw the standard. So what types of channels are we talking about that affect our graded potential? As the cell is repolarizing, as it reaches a minus 50 to a minus 55, that hyperpolarization opens a particular type of channel. Do you guys remember?
Funny channel?
The funny channel. That's such a weird name. Not very scientific. So funny channels open. Primarily sodium enters the cell, though some potassium will leave through the funny channel. And then what happens just prior to reaching the threshold of our action potential? The transient. So this is actually another threshold, okay? It's a threshold for our transient T calcium channels. So that allows another flood of positive ions to enter into the cell. Right? Both of those together, the funny channels and the transient calcium channels, bring the cell to this point. And what do we call that? Threshold potential. Okay. What type of channels open to create our depolarization? Because this is our action potential. Yes, the long-lasting what? Calcium. So in our other 2 examples, the ones that I erased, the skeletal muscle and the cardiac contractile, that depolarization leg of action potential for both are voltage sodium. Okay? So this is our big difference, the exception to the rule. So this is going to be long-lasting voltage-gated calcium channel. Will open, yes.
That happens at the threshold potential?
That the threshold potential is what opens those. Okay? That's what is the voltage-gated. This is also known as fast calcium channels, as opposed to the slow that we'll see when we get to cardiac contractile cells. All right? So that's this depolarization arm here. And then that depolarization opens our voltage-gated potassium. Okay, and it's the slowly opening, slowly closing potassium channels here also that contributes to that depolarization. So voltage-gated potassium are closing, thus we're slowing down our loss of positive ions. And then at this point we open the transient calcium. So that's what this potassium is referring to here. Okay? So while they're still open, we're slowly losing potassium, and then they eventually close.